Okay, I reckon we'll start now. So I'll share my screen. So this week we're going to be covering epithelium, biotechnology, and integrative metabolism. So I'll start us off by covering epithelium. And once again, if you do have any questions, pop them in the chat and we'll answer them on the doc later on as well. So in the body, we have 4 tissue types. So we have epithelium, we have connective tissue, we have muscle tissue, and nervous tissue. And today I'll be covering epithelial tissue, and later on in the semester and next semester, you'll be covering connective, muscle, and nervous tissue as well. So epithelium is pretty much just the tissue lining the internal and external surfaces of the body, including the surfaces, the openings of internal organs, body cavity lining, and also blood vessels and lymphatic vessels. But blood vessels we call endothelium, so it's just a different term for the epithelium. So these characteristics of the epithelium, they differ based on where the epithelium is found and what it needs to do. For example, epithelium you find in the throat will be different to the epithelium you find perhaps in your small intestine because they need to perform different functions. So the key functions of epithelium is protection. So that's, for example, your skin, secretion, the small intestine and trachea, absorption, and transportation. And the different structures of epithelium will be covered later, but there are 4 main key functions. In general epithelium, there is very little space between the cells, and they have strong intercellular junctions, so that's how the cells are attached to each other. Epithelium are oriented and polarized, so the apical surface differs to the basal surface, which I'll cover later. Epithelium sit on the basal lamina. Epithelium don't have blood vessels in them, so there are capillaries in the connective tissue underneath, which provide nutrients to the epithelium, but this isn't in the actual epithelium itself. And epithelium also have a very high regenerative capacity, so if they're injured, they're able to regenerate. Okay, so now we're having a look at some surface specializations on these epithelium. So on the base of your epithelium, it's composed of collagen type 4, proteoglycans, and glycoproteins. And the base of the epithelium functions to adhere the cell. It binds it to the plasma membrane at the bottom. It sequestrates growth factors. And normal epithelial cells can actually never cross the basal lamina. So cancer cells can do this, but normal epithelial cells don't. So when you're looking at a histological slide, if you see epithelial cells that have crossed the basal lamina, that should be raising a bit of a red flag to you because that shouldn't be the case. So if you have a look at the base, we've got 2 basal specializations that you need to know. So the basal lamina is otherwise known as the basal membrane, and that's what the epithelial cells attach to. So it's actually created by epithelial cells, so it's made of the collagen type 4, proteoglycans, and glycoprotein. And more important are the hemidesmosomes, which are what is responsible for binding the epithelial cells to the basal lamina. So they link the cytoskeleton to the ECM. So if you see in this image here, if you see the little disc and the little linking, that's your hemidesmosome, and you just need to kind of know that that exists. So now we go to the top of the epithelium, the apical specializations. So I've got cilia, which were actually covered in your very first PSP. So they've got their 9+2 arrangement, if you remember, and they beat in a wave-like pattern. So they're important for propelling material along the surface. So if you see in this diagram here, we've got cilia, and also in this diagram, you can also see the cilia there. Next, we've got microvilli, which are very important. If you see in this image first, you can see those little tiny feather shapes that go there. That's the microvilli inside the lumen of this structure. So they're important because they increase the surface area of the structure because there's lots of fine little finger-like things which increase the surface area for absorption, and they are relatively non-motile. So they're not like cilia, they don't move around and propel material, they're just there for absorption. So they show up on imaging quite darkly because they contain actin. So they're also referred to as a brush border because you can see it on imaging and it looks a bit like a hairbrush. And then we have stereocilia, which contrary to how the name might suggest, the name makes you think that it's cilia and that it's motile, but it actually isn't. So they're very long microvilli, but they're only found in a few things, so like the epididymis and ductus deferens. So they're the 3 apical specializations that you need to know about. Next up, we have the lateral surface specialization. So this is pretty much how each epithelial cell connects to the other. So we've got our tight junction, which also are interchangeably referred to as the zonular occludens, and they're the most apical junctions. So they completely encircle the cell and they provide a seal that prevents the flow of material between cells in either direction. So material can't flow from cell to cell when you've got a zonular occludens connecting the two. Oops, I'll go back one. Oops, I'll stay there. So pretty much the two plasma membranes that are next to each other, they completely fuse, and there's no flow of material that can happen. Next, you have the belt desmosome, which is also known as the zonular adherens, which encircles the cell as well, and provides some adhesion, but it doesn't completely fuse the adjacent plasma membranes. Oh. Next up, we have the macular adherens, which are also known as spot junctions and also known as desmosomes. So these are thickening on the membranes of adjacent cells that you can see. They have a thin, dense line in the middle of the intercellular space, and they're a site of attachment between cells and their cytoskeleton. And then gap junctions do not have any fusion at all between the two membranes, and they're 6 subunits arranged around a central pore. These pretty much facilitate cell-to-cell communication. So you can have chemical or electrical signals that diffuse between them, and they are important for spreading action potentials. So when you learn more about nervous tissue, you'll learn a lot about gap junctions. They'll be what's used to spread the action potentials. I'm not sure if I have I don't have a diagram. I might put a diagram on the question and answer document as well, so you can refer to that later. So next we have the different epithelium subtypes. So how do you initially identify epithelium? From site to site, you've got different epithelium that looks and behaves differently. So it's important to know how to differentiate between them. So the number of cell layers, whether it's simple, which is one cell layer, or stratified, which is more than one cell layer stacked on top of each other, is how you can characterize epithelia. But also you've got the shape of the most superficial cell. So the cell that's closest to the apical surface, you can characterize your epithelia based on the shape of that. So you've got squamous, cuboidal, and columnar. If you have a look at this image, we've got our squamous, which are more wide than they're tall, and they kind of look a little bit like fried eggs there. You've got cuboidal, which are equally long and tall, and you've got columnar, which look very tall and quite skinny. So we've got simple squamous. This is the first membrane type that you need to learn about. It's very simple, one layer of squamous shaped cells, which enables rapid diffusion across the epithelium. So this is found in places where rapid diffusion is needed. Examples are blood vessels and lung alveoli, because you need materials to diffuse very quickly across both of those. The layer is very permeable because it's so thin, and it's just squamous shaped cells. So if you have a look at this image here, you can see the epithelial layer there. All right, next up we've got simple cuboidal. They're more spherical shaped nucleus, and they're found in places that need secretion and absorption. So you find them in kind of salivary glands, renal tubules, ovaries, respiratory bronchioles, all those sorts of places that need secretion and absorption. After that, we have our simple columnar, which is an elongated nucleus. If you see here, the nucleus is quite long, but we've also got a very elongated cell as well. And the nuclei are usually positioned towards the bottom of the cell. So if you see here, the nuclei towards the bottom and the nuclei towards the bottom here as well. So they're also used for absorption and secretion, particularly in the lining of the stomach and the intestines. Next up, we have our stratified squamous. So in a stratified squamous epithelium, as you can see, it's lots of layers of the squamous shape stacked on top of each other, and you can't really have diffusion across this epithelium because the gap junctions or any junctions are all located in different places, so it makes it very difficult for materials to diffuse across the epithelium. So it's lots of layers of squamous shaped epithelium, and it's often found in the lining of wet cavities, for example, the mouth and the esophagus. Initially, the cells are cuboidal and they're attached to the basement membrane, and then these cells in the basement membrane move to the surface where they lose their nucleus and become more squamous, if you have a look there. And then a subdivision of your stratified squamous epithelium is your keratinized stratified squamous. So this, if you can see here, this epithelium covers the outer layer of your skin, so your epidermis. So this is what you'll find on your skin. And the nucleus gradually condenses until it disappears, and then the cell's dead. So the epithelial cells accumulate keratin, intermediate filaments, which pretty much just makes your skin waterproof, and it is subject to abrasion, but because epithelium is so regenerative, it's not an issue for this. So if you have a look in K, you can see your intermediate fibers, they're your keratin fibers interlinked, and in G, you can see your epithelial cells. So these cells have very tight junctions, so they probably have zonular occludins, which cross-link to prevent anything from getting into the epithelium itself, and your keratin also protects it and makes it waterproof. Next up, we've got our stratified cuboidal, and this one is a bit more rare. So you generally see it as 2 layers of cuboidal, not 3 like this image suggests. It's found in the largest ducts of glands. So you'd see it in sweat glands, mammary glands, and salivary glands, but it's more rare. It's not one of the more common ones that we talk about. I'd say the first 4.5 are the most common ones that you'll probably be asked on in an exam. And next off, we have stratified columnar. So you can see here you've got your taller columnar shape, but there's 2 layers of them, so they're arranged. These are very rare in the body. You only ever find very small amounts in male urethra and large ducts of some glands. So they're not quite as high yield as the other ones that we've discussed. But in general, epithelium is quite a frequently asked question on exam, so it is important to know all the subtypes if you can get around to it. All right, next up we have transitional epithelium. So this epithelium is unique. It's only found in the urinary tract, and it's waterproof and expansile. So the waterproof nature of this epithelium is necessary because urine's quite toxic. If you have a look at the epithelium here, it does look a bit like a mesh between stratified cuboidal and stratified columnar as well. So if you can see, it just looks a bit weird. But pretty much the surface cells, so if you see the surface cells here, they're called umbrella cells because they're rounded and sometimes they contain 2 nuclei. I don't think any of the ones in these images have 2 nuclei, but that's something to look out for if you're wanting to identify transitional epithelium. And they change shape based on their state. So when the blood is relaxed, they look like this. When the blood is contracting, they look like this. So they're very important because they've got a lot of stretch. And yeah, they're only found in the urinary tract. Next up, we have pseudostratified, which is a bit of a tricky one because it looks like you've got multiple layers in your epithelium, but there's actually only 1 layer. All of the cells are attached to their basement membrane. So if you see here, every cell is attached to the basement membrane, even though it looks like it could be some, stratified columnar or stratified cuboidal shape. So only the tallest of the cells actually form the surface. Some of the cells terminate a little bit earlier, but you won't see them because they're not forming the surface. And they're found almost exclusively in the respiratory tract. Pseudostratified epithelium is usually ciliated. So if you see this one, this is a pseudostratified ciliated columnar epithelium. And that's often found in your respiratory tract and also in your male reproductive system. So that's all the types. And then there's just a very short note on your exocrine and endocrine glands. I know the lecture wants that you appreciate that both exocrine and endocrine glands are derived from epithelia, and that's pretty much all you need to know. But the epithelial wall can form a well. So exocrine glands we see secrete into a duct, and endocrine glands secrete into the circulatory system, not via a duct. So if you have a look at this, this image here shows some exocrine glands. They still maintain their connection with their surface epithelium. So if you have a look, here's one, here's your epithelium, and even though they're still forming glands, they're still maintaining their connection with the epithelium, which has formed the well for these glands. And I think that is the end of mine.
Hey guys, so I'll be taking you through biotechnology for biochemistry this year. And I just kind of wanted to, before we start, emphasize the fact that your lecturers really only expect you to understand the basic principles. So if we have a look at the next slide, May, you can see that these learning objectives really want you to understand the overall process rather than worrying about the nitty-gritty of everything. So as we go along, I'll try and explain which ones are really important to know and which ones aren't so important to know. So just moving on, we'll start with recombinant DNA technology. And this is essentially where we grab genetic material from 2 different DNA sources and we combine them. And buzzwords that you want to be looking for in an exam situation is anything relating to the production of proteins. So recombinant DNA technology is mostly used for the production of proteins such as insulin. So keep an eye out for that if you get an exam style question asking about that. So we're going to have a look at some important tools that are used in this technology. And firstly, you have restriction enzymes, which are enzymes that will cut your DNA sequence at specific sites. And more often than not, you'll be left with what we call sticky ends. So if you have a look in this diagram on the left, in the top left, we've got a kind of zigzag red line that produces sticky ends, which are where you get a few unpaired nucleotides at the end of the strand. If we just had a vertical line, that wouldn't be a sticky end. Anyway, so once these sticky ends are produced, we can use them to connect previously separate pieces of DNA using what we called ligase. So ligase is an enzyme which will covalently bond DNA sequences together. So if we have a look in the diagram in the top right corner, we can see we have sticky ends that have been separated using our restriction enzymes and then joined together with another strand using the ligase. Once these DNA fragments have been rejoined, we will find a vector, which is usually a plasmid, a virus, or artificial chromosome of some kind. And the key criteria for a vector to be appropriate for recombinant DNA technology include 1, having an origin of replication, 2, a recognition site for the restriction enzyme, and 3, a selectable marker such as antibiotic resistant genes. In terms of application of recombinant DNA technology, it's super common in things such as farming, so farming with a pH, which is where you would insert this vector into an animal embryo. So your offspring is transgenic and therefore able to produce the desired protein. Okay, let's have a look at the process now. So I've tried to break this down as simply as possible. So firstly, we will identify our gene of interest and isolate it, as we can see in the diagram on the right-hand side, that left part. Using restriction enzymes, we will cut the gene at specific recognition sites for both the isolated gene and our plasmid, and our vector. And this will create compatible sticky ends, which we will bind together using ligase. We can see in this middle part of the diagram, we now have a ring formed by the DNA ligase connecting our isolated gene and our plasmid together. And now we can insert our vector into the host cell down below via heat or electric current, effectively transforming the host cell. Then we'll add a selecting factor, usually an antibiotic resistant gene. And what this allows us to do is that any cells that do not take up the vector will be selectively killed off, which allows us to culture successful cells. Okay, comparing different vehicles for and mechanisms of vectors, we have 4 main ones that are discussed: bacterial, yeast, insects, and mammalian cells. And basically, key points to take away are that bacterial vectors are the quickest and they're also the most cost efficient. Yeast will give us the highest yield of vectors, so the most in terms of per number. And then mammalian are able to deal with post-translational modifications the best. So post-translational modifications are chemical modifications made to a protein after DNA has been transcribed to RNA and then translated into protein. But essentially, it's able to do this the best because it can handle larger proteins better. Just got this last row, so going back, looking at application, the key ones to take away here are just remembering that bacterial vectors are for prokaryotic and simple eukaryotic proteins, whereas complex eukaryotic proteins are mammalian. So I would just probably take away those 2 as the most important ones to know. Okay, on to polymerase chain reaction. So we're going, again, I've tried to break this down as simply as possible. So the purpose of PCR is to create massive quantities of a selected segment of DNA. A full cycle will double the number of DNA strands that you have. With these steps on the right-hand side, it's actually really important that you remember approximately what temperatures are required for each of these steps, because this is a pretty common exam style question. So first we have denaturation at 95 degrees Celsius, which will separate the strands of DNA. Next, we have a process of annealing where we cool the strands of DNA to 55 degrees Celsius, which allows the primer strands to attach to the 3' ends of each original strand. We then have a process called extension where we reheat it up to 72 degrees, as well as adding a heat-resistant polymerase known as TAQ, and this helps to synthesize a new strand for each individual strand. So TAQ is the preferred polymerase here because it works quickly at high temperatures, and also it will use the primer as the starting point, which we want. So let's have a look at a diagram. So here we have on the left-hand side, we've identified our sequence of interest, and we've denatured it by heating it to 95 degrees Celsius, separating the two strands here. In our annealing process where we cool it, we're adding our primers to the 3' ends. Now, what these primers will do is define the region of DNA that will be amplified. So in our extension process, you can see that we have a new synthesized strand of DNA being extended from the primer at the 3' end. And we can see at the end of the first cycle, we have 2 copies. At the end of the second cycle, 4 copies, third cycle, 8 copies, and so on. Okay, on to therapeutic cloning. So again, we're just looking at simply defining the concept and describing different approaches. For year 1 medicine, there will be 2 approaches to know. Let's have a look at the first one. So the first one is somatic cell nuclear transfer, otherwise abbreviated as SCNT. And we'll go through the process here. So we remove the nucleus from our recipient egg, as well as removing the nucleus from the patient's somatic cell. The patient's nucleus is then inserted into the egg, as we can see here in this diagram, and this egg will proliferate, forming a blastocyst, which is otherwise known as a fertilized egg. And then embryonic stem cells will be collected from the blastocyst and cultured into embryonic stem cell lines. So these cell lines can now be induced into any differentiated cell, which is a major advantage for a somatic cell nuclear transfer. On to the next one, we've got this second method is induced pluripotent stem cells, which is where we extract our somatic cells from our patient, and then a vector containing 4 gene coding proteins is then inserted, which causes the somatic cells to become undifferentiated back into a induced pluripotent stem cell stage. Just to remind you guys, these pluripotent stem cells can give rise to any cell in the body except for placental ones. So stem cells that can differentiate into any cells, including placental ones, are embryonic stem cells. Pluripotent do not differentiate into placental ones. Totipotent can give rise to any cell, and multipotent can only give rise to cells in their own lineage. Let's compare the two. So with pros, with somatic nuclear transfer, we've got reduced chance of immune rejection, which is really important, particularly in organ transplant surgeries. And so this will reduce wait times on the list, okay? But in terms of cons, we've got huge ethical considerations regarding the ethical concerns about creating or destroying an embryo. Whereas with induced pluripotent stem cells, we don't have these kind of ethical considerations by definition, but we can get a few more cancer-causing genes and a few tumor suppressor genes, which will lead to the development of cancer. Additional methylation is required, and there can be some epigenetic differences, which means that it's not always successful. Epigenetic differences means that essentially some genes can be modified over time due to the environment that you grow up in. And just before we move on, sorry, I'll probably just quickly summarize that we use these processes of therapeutic cloning essentially to help us understand disease, develop new treatments for disease such as cancer. So largely there's a very research and clinical aspect to it as well. Okay, so moving on to CRISPR-Cas9, again, we're looking at how it works in pretty basic understanding terms. So let's have a look. Okay, so starting off with gene therapy, unlike other technologies, what makes gene therapy unique is that it requires modification of cells that you already have, that already exist, and tissues in the patient. So a vector will deliver new genetic information to the patient. So rather than correcting something that's wrong or replacing the DNA sequence entirely, we're actually adding this new genetic information. Okay, so the process is essentially where we identify two defective alleles in a patient. This normal allele is inserted into a virus vector to create recombinant DNA. This virus that contains the recombinant DNA will be inserted into the patient's somatic cell. And hopefully through this process, the normal allele will produce the functional proteins that are required. So yeah, we can produce functional proteins, but unfortunately at this current stage in time, the duration is not particularly that long as your introduced allele may degrade over time, as well as the issues with immune rejection that come from inserting a viral vector. So you can get a response from either your innate or adaptive immune system. And then if we incorrectly edit and we accidentally add a dangerous form of DNA, a dangerous sequence, we could lead to the formation of cancers. So onto CRISPR-Cas9 gene editing, we basically use CRISPR-Cas9 kind of in research related to cancer, virus infections, genetic diseases, detection of pathogens, those kind of things. And how it works is your Cas9 will bind to the segment of RNA that matches the target RNA. So we're identifying the target DNA. Your Cas9 will then cut the DNA here, and essentially the sequence can be deleted and the cell repairs the DNA. So it involves gene deletion and addition with somatic cells, but we are in the early stages of kind of CRISPR-Cas9 technology. So we've got a few weaknesses, one being we can edit the wrong gene, or we can cut in the incorrect place, which can be disastrous if we lead to the formation of cancer. So largely in an early kind of stage, but we can see the kind of potential it has moving into the future. Okay, so I'll move on to integrative metabolism now, looking at a focus on the interrelationship, just the next slide, right? Yep. Between carbohydrates, lipids, and proteins in the liver, and explaining energy sources used in the fed and fasting states. So quite simply, our liver, just remember that it's used to process fats, carbohydrates, and proteins. It will synthesize and distribute these lipids, ketone bodies, and glucose for other tissues in your body. It will convert excess nitrogen to urea, which is super important. And in the fed state, so if you've had some food, your liver will store this energy, but when you're hungry and you're fasting, it will break down this energy by accessing your stored energy reserves. So in the fed state, so just remembering that the liver will process fats, carbs, and proteins, will have an increased formation of glucose 6-phosphate because there's more glucose in the blood. And glucose 6-phosphate will trigger the start of metabolic pathways such as glycolysis and the pentose phosphate pathway. So that's why glycolysis and pentose phosphate will increase, as well as the fact that glucose 6-phosphate can be converted to glycogen which is a form of energy storage. So we also get increased glycogen synthesis in our fed state. Glycolysis is really important here because it will extract energy from the glucose by breaking it down into 2,3 carbon molecules that we call pyruvates. And the pyruvates that are formed will help trigger fatty acid metabolism because they will be converted into acetyl-CoA, which can then be converted into fatty acids. So that's why we have increased fatty acid synthesis. And because of this, the fatty acids and the glycerol that you obtain from your diet will then be transported to the adipose tissue via very low density lipoproteins, so VLDLs, and stored as triacylglycerides, which is why we have increased triacylglyceride synthesis. Any excess amino acids will be degraded as they can't be stored in the body. So with our amino acid metabolism, which is our proteins, will have an increased rate of amino acid degradation. However, the carbon skeletons that arise because of this degradation will be recycled, converted to pyruvate, and then into acetyl-CoA, again, contributing to increased fatty acid synthesis. So yeah, just taking away that these are all interrelated, but most of these processes will increase in our fed state. In contrast, our fasting state is quite a bit more, quite less complicated, sorry. So the liver, remember, needs to make energy available from its storage. So our adipose tissue, where our triacylglycerides are stored, will be mobilized and transported to tissues where the fatty acids are oxidized for energy. And the glycerol from the tags in adipose tissue will also be converted into glucose via gluconeogenesis. So we've got release of fatty acids to supply other tissues. Then our liver will convert the amino acids that we have into other nutrients, primarily glucose. And then the glucose will be exported to other tissues. Acetyl-CoA from our amino acids and fatty acids will be used to make ketone bodies for the brain. And acetyl-CoA will be produced in the beta oxidation of fatty acids in the liver. So yeah, we can see brains will use ketone synthesize. Our glycogen will also be broken down into glucose, and these all help to provide energy for our body. I'll now go and show you guys some super high yield tables. So these tables are really important because they're not necessarily covered in your lectures, but they're often brushed over in your tutes, but will most definitely be tested in your exams. I can guarantee you that. So let's have a look. So this is a table outlining the energy sources that each major component of the body uses. So mainly the heart, brain, red blood cells, and skeletal muscles. And these will be the ones that you need to know for your exam. So your key takeaways are your cardiac muscle, the heart, will use primarily fatty acids from blood circulation. And then when the fatty acids aren't available, it'll turn to glucose and ketones. So fatty acids first, followed by glucose and ketones. Just important to note that the heart can't store these fatty acids, so it constantly requires sufficient fuel in some form or another. Looking at the brain, it's primarily blood glucose. Again, it's not able to store it, so it needs constant input. If it's unable to receive blood glucose because your blood glucose levels are low, the brain will respire ketones in the body because these ketones can cross the blood-brain barrier that other energy sources can't. Red blood cells, the key points to remember are it's always glucose, and that's because red blood cells use anaerobic respiration as its main pathway because red blood cells do not have organelles. So lots of glucose is needed because anaerobic respiration won't yield much ATP. So you can kind of see why having regulating your levels of glucose in the human body is super important because we can't get a lot from anaerobic respiration as opposed to aerobic respiration. Lastly, with skeletal muscle, this is quite general. I go into this in a bit more detail in the next slide, but generally we will use lipids and then we use proteins if our lipids aren't available, but I'll go into these in more detail in the next slide. When you're ready, May. Yeah. So looking at skeletal muscle in our first row, at rest, we will use fatty acids, but during our bursts of activity, so for example, sprinting at the start of a race, that'll be a buzzword you'll see in your exam. So short sprint from 0 to 30 seconds. We will use creatine phosphate. With moderate activity, it'll mainly be glucose followed by fatty acids and ketones. In starvation, proteins are broken down as a last resort. And just revising from the last slide, red blood cells will always use glucose. The heart will prioritize fatty acids, then glucose and ketones. The brain will prioritize blood glucose followed by ketones, as these can cross the blood-brain barrier. Okay, on the next slide, we've got this, I'm introducing you guys to the topic of the Great Metabolic Race. And the Great Metabolic Race is essentially looking at, if I was running in a race, what energy sources would I use to supply myself, okay? And this is, you'll definitely get a few questions relating to this on your exam. So yeah, I'm aware you guys have done a workshop. We'll do something to revise this right now. So the key point to take away from this diagram is at 30 minutes, your carbs and fats swap over. So your priorities change. Let's have a look at the next slide. Okay, so at rest, before our race, our fatty acids are our main source of energy along with ketones in the liver. They are slow, but they release lots of energy, which is a good thing because that'll last us for longer. 0 to 2 minutes can be broken up. So we've got our short burst from 0 to 30 seconds where we will use our ATP reserves and creatine phosphate, like I mentioned before. And then for the rest of those 2 minutes, we will probably use free glucose in the blood, as well as carbohydrates. In our first 2 minutes, again, our fatty acid use is decreasing while our carbohydrate use is increasing. And then at the 5-minute mark, again, it'll mostly be carbohydrates, so around 85% compared to 15% of fats. So we start to use our stored glycogen and blood glucose with muscle glycogen converted to lactate here. At our 30-minute mark, this is where we have approximately equal we're using an approximately equal percentage of carbs and fats. But after 30 minutes, fatty acid metabolism is our main energy source. So this will be more than glucose metabolism. So we're releasing more energy, but more slowly. At around 8 hours, proteins are really the only things that are left. So your key takeaway here is that in periods of extreme starvation, the body will break down proteins, but it is simply a last resort. And this will be part of, I believe, the glucose alanine cycle, which hopefully you guys would have covered. And then just the last row there, just summarizing that. Now, whenever you get a sprint or a short burst, so anytime you see that word in an exam question, you should be thinking creatine phosphate. So there's a small supply of these in cells, such as ATP reserves. They're in limited supply, but they're really good for sprinting. So that's the main thing to remember there. Okay, so that's kind of the metabolic race covered, as well as biotechnology. There is a lot of information to kind of digest. But if you guys have any questions at all, please message either May or myself, and we'll put this recording up on YouTube as well for you to watch through it again, if need be. And of course, feel free to access the slides as well